Cell polarity is the fundamental concept of physiology. In this video, I'm going to tell you what is cell polarity, its maintenance, and functions. Hey everyone, welcome back to HM Learnings. I'm Harshita, the creator of HM Learnings, where students come to clear their concept and to get the study material. Make sure you subscribe my channel. So today we are going to look with a very basic topic of journal physiology, that is cell polarity. So before going into understanding what is cell polarity, we will first understand the meaning of polarity. So basically the polarity word has been come from the word pole, and pole means the two ends or the extremes or the extreme points of something. Like we use pole in the case of the earth, north pole, south pole. So basically to see that these are the two ends, these are the ends or extreme points of entity, we use the word pole. And polarity means what? Polarity means that the ends which we are referring, they are actually opposite, or not exactly always opposite, but they are different from each other. So this polarity word is not exclusively used for the cell only. It is being used very commonly in the physics for the south pole, north pole. There also we discuss the polarity in the magnetism. Then in the case of the chemistry also, if you know about the polar covalent bonds, non-polar covalent bonds, in the case of the heart also the word dipole is being used, and also in the cytoskeleton this word is being used. So we can understand this polarity word by taking an example of a circuit. So we are having a circuit, and we know in the circuit we are having a positive end of the battery, and this positive end of the battery is linked with a resistor, suppose, and then the negative end of the battery is also linked with the another end of the resistor. So you can see that the current flow direction is this, suppose. So basically if you see this battery, one end is positive and the another end is the negative. So these are two poles or two ends of an battery, and they are showing the polarity in the way because they are actually uh, opposite from each other. The plus is going to be the electron deficient, and this negative is going to be the electron rich. So that is how whenever you consider the flow of the current, you will always see that you will find the difference in the terms of the electrons, and that is how the two ends are going to be marked as opposite and different and having the polarity. So in the case of the heart also, the main vector which is showing the direction of the current is this, with the positive end here and the negative end here. So this is also sometimes referred to as a dipole, that is two poles which are having the polarity. Or we can say, if we take the example of the polar covalent bonds as such, in the case of the, if you see the bond between the this thing, the water, you can say. So in the water you can see that between the oxygen and the hydrogen, this is the bond which is a polar covalent bond. So oxygen is something which is more electronegative. It is having, it is going to tend more electron, it is going to pull more electron towards it, and that is how it is more electron rich as compared to the hydrogen. So it is having a partial negative charge, and this is having a partial positive charge. So then they are actually having a dipole or showing the polarity. So what I meant to say is that polarity means the 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 ends which are having the opposite or different properties from each other. And similarly the cells are also having the polarity in the way because they are showing the asymmetric distribution of protein, organelles, cytoskeleton, as well as the cytoplasm. So if you consider any cell, you will see that it's not always the protein, one protein. If you take any example, suppose not actin is actually a cytoskeleton. So if you take any other protein example, you will see that that protein is not going to be present uniformly throughout the cell. Some portions of the cell are having more concentration of that protein, some portions are having the less concentration. In the case of the organelles also, if you remember that the nucleus, immediately to the nucleus there is presence of endoplasmic reticulum, and then there is Golgi body. So again, it's not, they are actually present close to each other, so they are showing the asymmetric distribution. Cytoskeletons also, like the actin or the microtubules, especially during the cell division, they are showing the asymmetric distribution. They are having the role in making the mitotic spindles. So that is again, so it's not only the other parts, but cell is showing the polarity in terms of the distribution of the protein, organelles, cytoskeleton, and the cytoplasm. So now the next question which comes to mind is, does all the cell exhibit the polarity? The answer is yes, almost all cells exhibit cell polarity, but most commonly we take the example of epithelial cells and the neurons to understand the cell polarity. So today we are going to take the example of epithelial cells to understand the polarity, and in that also we will going to take the example of simple epithelial cells of intestine and proximal renal tubule. So before going to understand what we mean by polarity, and to discuss it in more detail, we will see what is the general layout of epithelial cells to understand the structure of it. So if I draw one epithelial cell, so this is a simple epithelial cell. So simple epithelial cell means there is a single layer of epithelial cells which is lying over the basement membrane. So this is the basement membrane. This is a single epithelial cell which I have drawn. So now you will see here that this epithelial cell is having this membrane. So if you consider this as an intestine, you consider this as the lumen and this as the connective tissue, or the other side of the small intestine. So this is the side which is facing the lumen of the intestine is the apical side. Okay, the two sides lateral, these are the lateral sides. This and this are the lateral sides of the cell, and the below side is the basal side. The side which is attached to the basement membrane is the basal side. Okay, so this is, now this I have drawn a single diagram, single epithelial cell. Now I will draw many epithelial cells to understand what is the structure of the epithelial cell. So we are having another epithelial cell also, like this is one epithelial cell, this is again, this is epithelial cell, and this is again. So you will see here that this epithelial cell, this is the apical membrane of the epithelial cells. These are the lateral membranes of the epithelial cells. Okay, and these lateral membranes are the membranes through which it is attached to the neighboring epithelial cells. Okay, so these are the lateral membranes, and this basal membrane is the membrane through which it is actually attaching to the basal lamella. Okay, so these are the membranes. So this is a 2D structure. So in the 2D structure, you consider it as a rectangle which is having one apical side, okay, two lateral sides, and one basal side. Okay, but in reality it is not 2D, it is actually 3D. So there will be epithelial cells which are present in front of it, and there will be epithelial cells which are present back of it. So that means if I draw here like one epithelial cell like this, okay, and another epithelial cell like this. Okay, so from this you can say that there are 4 lateral sides. Okay, so it is like a cube. So cube, if you know, it is having the 6 surfaces. So in epithelium also there are one apical side, 4 lateral sides, and 1 basal sides. So in case of polarity, what we are seeing is that like the apical side of the cell, or more accurately we can say, apical means the apical membrane or apical surface of the membrane is having the proteins, is having the morphology which is different from the basolateral sides of the epithelial cells. So collectively the lateral and the basal side of the epithelial membrane is called as in basolateral. So basolateral is having different morphology as well as different protein composition as compared to the apical surface of the epithelial membrane. So now we see this diagram to understand further. Okay, so you can see here, this is what I have also tried to draw. So this is the epithelial cell. one epithelial cell. Okay, I'll use the pointer. Okay, so this is one epithelial cell. Okay, and this is another epithelial cell. So this is the apical side of the apical membrane of the epithelial cells. Okay, and this is the lateral side of the membrane. So this lateral side is showing the attachments to the lateral neighboring cell by the help of the intercellular junctions, as well as it is showing the attachment to the basement membrane again with the help of the intercellular junctions. Now these are the two cells, that is, this is a 2D structure. If you see behind it also, there is one more layer of the epithelial cells which is being shown, and likewise also there will be one more layer of the epithelial cells which can be drawn in front of it. So from this you can say there is one apical surface, one basal surface, 1, 2, 3 and 4 lateral surfaces of the epithelial cell. So now in this diagram you can see that the apical surface of the cell is different from the basal side. So the apical side is thrown into the microvilli. The membrane of the apical side, if I draw the diagram, you can see that this is the apical side. The apical membrane is being drawn into the finger-like projections which are called as in microvilli in the case of the intestine epithelium. Okay, and in the renal proximal tubule, whereas a basal side and the lateral membrane, they are having the basal infoldings. Okay, and they're having interdigitations on the lateral side. So all of these are actually increasing the surface area of the epithelial cell, but the structure which they are forming is morphologically different and protein wise also different from each other. That is one way in which they are showing the polarity or the differences. Another thing which is there is a protein composition. So in the protein composition also, if you consider it as a lumen of intestine, you will know that there must be presence of sugars and peptides, everything in the lumen, and to digest them, the membrane bond enzymes are present in the microvilli. In the membrane of the microvilli, those enzymes are being present. So they are going to hydrolyze these products, and as well as there will be presence of the transporters which are going to take them inside the cell. So by these transporters, these sugars and peptides, after getting hydrolyzed by this membrane bond enzymes, enter inside the cell. Now these transporters are going to be different from the transporters which are present on the opposite, the basal side of the membrane, which will help the transport of this molecule from inside the cell to directly into the blood. Okay, so now they will come into the blood. So this is blood capillary in which they are going to be entered. So the kind of the proteins which are present here in the form of enzymes, in the form of transporters, are different from the enzymes, are different from the proteins which are present on the basal side. They are helping in the transport in the opposite direction, that is from inside the cell to the outside of the cell, whereas they are taking the material from outside the cell to the inside of the cell. So the protein composition is different, and that is how the cell polarity is directing the transport of the molecules from the lumen across the epithelium to come into the blood. Okay, now apart from this we have taken the example of the epithelium which is having the absorption, the function, like in the intestine and the renal proximal tubule. There are other cells also, like if we consider the other epithelium also, if we consider the case, suppose this is a capillary and we are talking that there are simple squamous cells which are lining the capillary. Okay, and these are the nucleus of those cells. So this is the apical side of those cells, little bit of the lateral side, and then the basement cell. So here there must be the proteins which are attaching them to the basement membrane. There must be proteins on the lateral sides which are attaching them to their lateral neighbors, and there are the surface antigens. Okay, There are surface antigens which are helping in the communications or the recognition between the cells, as well as in the case of the endocrine cells. Okay, so there is a hormone in the blood and there is an endocrine cell. So to detect that hormone on the apical side of the cell, there will be presence of the G protein coupled receptor, or a receptor which is exclusive to that protein or to that hormone. So that is how the apical membrane is showing the different protein composition as compared to the lateral and the basement membrane. The lateral and the basement membrane are having the proteins which are attaching the cell to the another cell, or attaching the cell to the matrix. So in this diagram you can see that they are showing the tight junctions between the cells. This is the diagram of a tight junction. Okay, then they're also showing the macular adherence, the gap junctions, everything. These things are not present on the apical side. Only structure which is present on the apical side is a microvilli. Then there are other proteins which are present on the basement membrane. So this is what we have already seen. Ciliated epithelium is being present in the fallopian tube and the trachea also, where the apical side of the epithelium is having the cilia, which is helping in the transport of the gametes and the particle. Then there is specific channels or receptors also for the cell signaling. For example, neurons. So neurons, if I draw the diagram, we know that this is a single cell. So in the single cell, the one part is drawn into the dendrites. So they are having the different types of the channels or the receptors so that they can detect the presence of the neurotransmitter. Then there is the axon which is there, and then finally the synapse. So they are having the different proteins on different areas which are helping in the transmission of the signal, showing the polarity again for the signal transmission. The basolateral membrane are having the basal infoldings and the lateral interstitials. They are going to increase the surface area, and also the presence of sodium potassium pump, proteins of intercellular junctions, which we have already seen. So we will talk about the sodium potassium pump in this diagram. So you see that basically this is the lumen of the intestine, this is the intestinal cell, and this is the interstitial fluid, or you can say basically the opposite side of the cell. So this is the apical membrane which is facing the lumen. So in this membrane you will see, in the lumen you can see there is glucose which is present, which needs to be transported, and there are amino acids also which needs to be transported inside the cell. So there are specific transporters which are present for the transport of glucose, for the transport of the amino acid, okay, on the apical membrane, and these transporters are not present on the basal membrane. And this is how the polarity is helping in the energy efficiency. The enzymes which are not required, the proteins which are not required on the apical membrane are being present on, not on the apical membrane, but the proteins which are required on the apical membrane are present on the apical membrane. So in this way they are showing the energy efficiency. Now once the glucose has entered inside the cell, another transporter which will help its transport from out inside the cell to the outside is being present on the basal side. And the very important one, which is exclusively found only on the basolateral sides of the membrane, is the sodium potassium pump. And this, the number of the sodium potassium, as I said, the basal infoldings and the lateral interdigitations also going to increase the surface area of the lateral and the basal membrane. They are actually increasing the number of the sodium potassium ATPase pump because it has a role in the secondary active transport. So this is going to make sure that against the gradient there could be a transport of the glucose and the other essential another ions or the compounds inside the cell. Okay, so now we can come to the next topic, that is how does the cell polarity is going to be maintained. So as I said that some proteins are present on the apical side, some proteins are present on the basal side, some on the lateral side, but how does this polarity is being achieved? This is being achieved by the trans-Golgi network. So you know that the secretory proteins are being synthesized by the trans-Golgi network, where the synthesis of the protein is going to happen. in the ribosomal endoplasmic reticulum, then the packaging will be in the Golgi apparatus. So after that, there is something called as a sorting, where the proteins, according to their destination, whether those proteins are supposed to be present on the membrane or inside the cytoplasm or inside in some other organelle, the sorting is going to be happen. So this topic is, it's a very detailed topic which requires another video. So you just remember that there is a sorting of the proteins which is being done by during their packaging, during their transportation. So those proteins are going to be like the proteins which are specific on the membrane, they are going to be sent to the membrane. In that also, the proteins which are integral proteins, which are which are present on the outer side of the membrane or the inner side of the membrane, that much sorting is also being done. So every sorting is being done by using this trans-Golgi network. Another thing is the tight junctions and the other types of the junctions, which are ensuring that between the cells there is a limit of the paracellular diffusion. Now what does it mean? So we will look in this diagram. So you know that in the lumen, the concentration of glucose is going to be high initially, and the glucose concentration is going to be low in the cell initially. So basically there is a a gradient which is being there, but after some time what is going to happen, that the glucose is going to be high inside but low outside. So here what will happen, against gradient there will be secondary active secondary active transport of glucose from lumen to the intestinal cell. Now there is presence of tight junctions between the cell, and this ensures that glucose will transport only via this pathway across this membrane, not via this pathway. Okay, not via this pathway, through this paracellular transport. Because if it transport by this way, what will happen, by diffusion the gradient will remain the same throughout this environment. So ultimately the glucose absorption could not happen as it should be. So it is going to limit the paracellular diffusion so that the gradient could be maintained and the transport could happen efficiently. Another thing is that it is going to limit the diffusion of the proteins inside the membrane. So inside the membrane also there is movement of the lipid, there is movement of the proteins also, that is also going to be limited. Then cell polarity, what does it function? So I already talked about this function, got about this function, and also about this function, that it helps in the recognition by specific antigens, which is important for the immune response, also for the endocrine responses, because of the presence of the specific type of receptors on the apical side and on the basal side. So this receptor is also being this function is also being covered, and also about the and also it is important for the development and the differentiation. Okay, so I think there are some points which I left here. Okay, this is already clear. Now intracellular junctions ki proteins are also present on the basal side and the lateral side, and there are various functions of the intracellular junctions which are going to be covered only in the video on the intercellular junctions, but briefly you can say that they are going to adhere the cell to each other and to the matrix. They assure that there is no detachment of the cells when external force is applied to them. They are going to perform the function of mechanotransduction, and sometimes with the help of the gap junctions, like in the cardiac muscle and the smooth muscle, they allow the fast signaling between the cells as well. So this is all about the polarity. In this video we have talked about what is cell polarity, asymmetric distribution of the cytoplasm, organelles, cytoskeleton, and the protein. And in that we have talked about exclusively about the epithelial cells, little bit about the neuron, and the type of the polarity which we have discussed is the apical-basal polarity. So if you like this video, please like, share and subscribe the channel. If you have any doubt, do let me know in the comment section. Till then, keep learning.